这个这样。 Good afternoon, Rajasekharam. Hello.
Good afternoon, sir.
Good afternoon, Rajasekharam.
Good afternoon, sir.
Good afternoon, Dr. Ruth Garu. I think you are ready now with the biodata of Dr. Rajasekharam. Hello, Ruth Garu.
Sir,
are you
good
are you
afternoon, sir.
yes, good afternoon. Good afternoon.
Ready, sir.
Okay, okay.
Biodata.
Okay, it is with you now. Biodata is with you.
Biodata is with me, sir.
Okay. So Rajasekharam,
Sir, today is our 20th session of the CIPRES graduation readiness program, sir.
yes, ma'am. Yes, ma'am. I think Rajasekharam has joined. I think he has joined.
Join ayyanatla, sir. Join ayyar, sir.
Okay, okay.
Join ayyar.
I think, shall we wait for a few more minutes?
Start చేద్దాం, sir. ఇప్పటికి 4:12 అయిపోయింది.
4:12. I think we'll start by 4:15.
Start, sir.
4:15.
Start, sir. Okay, sir. Okay, sir.
Hello.
Sir, good evening, sir.
Good evening. Suraj Lakshmi, shall we start?
Start చేద్దాం, sir. Yes, sir. Yes, sir.
Okay. Dr. Rajasekharam has joined.
Yes, sir.
Sir,
Okay.
it's joined, sir.
Yes. Okay. We can start the program now. Good
Yes,
afternoon to all of you. I am Dr. Srinivasulu, Associate Dean, working at College of Horticulture, which is a constituent college of Dr. Rajasekharam Horticulture University. I take this opportunity to extend a hearty welcome to today's guest speaker, Dr. T.Rajasekharam, senior scientist working at Citrus Research Station, Tirupati. Actually, in today's program, Dr.K.Gopal Regist has to deliver the talk on this topic, budwood transmissible disease indexing in citrus. Because of some other important engagements, he will not be able to attend for this program. So on his behalf, Dr. T.Rajasekharam will be delivering the lecture on the proposed topic. That is a very important topic, budwood transmissible disease indexing in citrus. I extend hearty welcome to Dr. T.Rajasekharam garu for this program. I think this citrus graduate readiness program, which is a unique program that is being organized by Dr. Rajasekharam Horticulture University for the benefit of the final year students of both horticulture and agricultural universities. So far, 19 lectures have been completed, and this is the 20th lecture, guest lecture, that is being programmed for the benefit of the students. So far, many eminent speakers have been delivered their lectures on important topics. And in today's program also, an important topic in citrus will be dealt by Dr. Rajasekharam garu. As I said, this program is a unique program. I request all the student participants and also the faculty and other members who are participating in this seminar. I think this lecture, I once again invite them all to this program. This 20th program also consists of a quiz program, which consists of, I think that program will be, details of that program will be given after the lecture by T.Rajasekharam garu. It consists of some questions. I think students have to answer for that one. Students who have scored highest marks will be given certificate at the end of the citrus graduate readiness program on the final day. So in this today's program, I take this opportunity to once again welcome Dr. Rajasekharam garu to deliver his lecture on this budwood transmissible disease indexing in citrus. May I now request Dr. Ruth Banerjee garu to read out the biodata of Dr. T.Rajasekharam garu for the information of the today's participants.
Good afternoon to all of you. Now I will read out the biodata of the speaker. Dr. T.Rajasekharam received his PhD in plant pathology with specialization in virology from the University of Agricultural Sciences, Dharwad. He currently serves as a senior scientist in plant pathology at Citrus Research Station, Tirupati, and provides leadership to the research, extension, and training programs in plant pathology in horticultural crops. He has 11 years of research experience on plant viruses in horticultural crops. As a plant virologist, he continues to work on viruses of fruits, vegetables, and ornamental crops. His main research interests include molecular characterization and diagnosis of plant viruses, control of viral diseases of horticultural crops, and the development of environmentally friendly and sustainable integrated pest management strategies for reducing the impact of viral diseases, and also development of virus resistant varieties or hybrids in vegetable crops. And he has guided 4 MSc Horti and 1 PhD students in the Department of Plant Pathology and Horticulture. And he published 25 research papers in international and national journals. He has completed 3 projects on different aspects of citrus. Thank you, sir.
May I now request Dr. Rajasekharam to kindly start delivering the lecture on this budwood transmissible disease indexing in citrus. Dr. Rajasekharam.
Thank you,
Please.
sir. Sir, I'm audible, sir?
Yes, please. I think volume, can you increase a little bit? Yes.
Now it is okay, sir?
Yes.
Good evening, one and all. Thank you for giving me this opportunity. You know, citrus is one of the important crop. It is infected by several viral diseases, bacterial and fungal diseases. Among these diseases, this budwood transmissible diseases are very important. Why? Because in case of sweet orange, we normally graft rootstocks with the budwood from the allied plants. Once the mother plant is infected with the diseases, these budwood transmissible diseases, the progeny also will be infected with these diseases. So once whatever the plant material with these diseases is supplied to the farmers, they definitely. Madam?
Voice chala low ga vastundi.
Now it's okay, madam? Hello? Is okay, madam, now? Is audible?
It is okay, Rajasekhar.
Now it is okay, madam?
Okay, Rajasekhar.
Okay. Citrus is the third largest, India is the third largest producer of the citrus fruits after China and Brazil, producing around 14.07 million metric tons. Further, it is contributing around 8.9% of the total world citrus production. This is one of the important crop in India. Citrus is third most important fruit crop in India after banana and mango, plays a very important role in economy of the country. Among the citrus, mandarins occupies 43% share in total production. It is followed by the sweet oranges, that is Mozambique, Southwoody, Malta, Amrin, Jaffa, and pineapple, etc. Other limes and acid limes are also contributing around 26%. Similarly, rest of the fruits, including grapefruit and pomelo, contributes around 5% of total citrus production. These are the main groups of the citrus fruits producing in India. So in India, around 1.3 million hectares citrus fruits are growing with the production of the 14.07 metric tons. The productivity is only 10 metric tons. In India, Andhra Pradesh ranks first in area and production of sweet oranges and contributes 61.34% share in sweet orange production. So acid lime production occupies second place after Gujarat. These are the two most important citrus fruits produced in Andhra Pradesh. Though there is several fold increase in the area in citrus cultivation as well as citrus production in India during the last 5 decades, but there is very low productivity. It is around 10 to 11 tons per hectare. as compared to 28 to 30 tons per hectare in Brazil and USA. In Andhra Pradesh, sweet orange productivity is around 10 metric tons during 2000, and it is gradually increased to 21.52 metric tons in span of 2 decades, but it is below the potential productivity of 30 metric tons per hectare. One of the main reasons for this poor situation is infections caused by the several virus and virus-like pathogens that affect citrus plant health, productivity, fruit quality, and finally causes the citrus decline. And use of uncertified poor quality plant materials also contribute to this poor yields. In horticulture crops, more than 60% infections are attributed to virus, viroids, and phytoplasmas. These pathogens are systemic, and particularly these are more traded vegetable propagated crops like citrus, banana, and tuber crops. In case of citrus, there are around more than 160 diseases are reported in citrus. Among these diseases, the fungus, bacteria, nematodes, and virus pathogens are causing. Fungus, these mainly the causes are root rots, leaf spots, and fruit spots. These are the different diseases caused by the fungus. Bacteria causes the citrus canker. Nematodes, citrus nematode is also one of the important pathogen. It causes the slow decline in citrus. Whereas in case of virus and virus-like diseases, there are total more than 50 these systemic infections occur on citrus. And these virus diseases are bud and graft transmitted diseases. And these diseases, in viruses, there are 8 groups of viruses are infecting the citrus. Among these, Clastro, Columnoviridae, Flaxiviridae, Bromaviridae, these are the different groups of the viruses infecting the citrus. And 5 different viroids are infecting citrus crop, including citrus exocartes. The 4 different prokaryotes are infecting. Among these, Xylella, which is caused variegated chlorosis in Brazil, and Spiroplasma is also causing citrus stubborn in US and Mediterranean region. These both pathogens are not present in India. Whereas Phytoplasmas, there are 5 groups of Phytoplasmas infecting citrus crop, and fastidious phloem inhibiting bacteria is Candidus cibrobacter asiaticus. This is also one of the important pathogen infecting the citrus and causing the citrus greening disease. And these diseases are reported in India. In India, 16 such virus and virus-like pathogens are infecting citrus crop. Among these, 5 pathogens infecting the citrus and causing the significant yield loss. That includes citrus urticaria, ring spot, yellow mosaic, citrus greening, and exocartes. Either as a single infection, as mixed infection, these pathogens plays a significant role to cause citrus decline in India. The symptoms in photos are highly conspicuous, but in the field condition, these are not clear due to low concentration and uneven distribution in the plant. Sometimes these viruses and virus-like pathogens confined to the few parts of the plant, especially in roots and twigs. These concentrations are accumulated on those parts, and those symptoms are not conspicuous, as this citrus, is a woody plant. And especially in case of citrus greening, symptoms are more conspicuous during the winter months. During the hot summer months, the symptoms appearance generally must, and it is very difficult to identify this pathogen during the summer months. Therefore, there is a need for development of proper diagnostic tools for the diagnosis of these diseases. There are numerous diagnostic tools are being used for the virus detection, starting from symptomatology to microarray, having different sensitivity in terms of detection limits. These techniques have their own advantages and disadvantages and limitations. Choice of techniques that can be used by the scientist depends upon the type of experiments, availability of resources, and availability of the human resources. These are all governs the what method that can be used for diagnosis of the diseases. So among these diagnostic methods, there are field diagnostic and laboratory diagnostic methods are there. Field diagnostics is mostly based on the symptoms produced on the plant parts. But in case of citrus, these symptoms are not conspicuous, and these symptoms have very limited value in diagnosis of these diseases. And in case of laboratory, biological indexing, electron microscopy, immunological and nucleic acid based techniques are being developed since several decades. Electron microscopy is a direct method. So directly we can see the viral particles, the electron separations. Immunological methods are ELISA, immunosorbent electron microscopy, blood immunobinding assay, tissue blood binding assay, and leaf lateral flow deep method. These are the immunological methods. Nucleic acid methods include the PCR, real-time PCR, microarray.
Rajsekhar garu.
Madam?
Screen, share the screen.
Sir, slide is
Share
not.
chesam madam.
Slide is not visible.
Already shared madam.
They
But
shared.
it is not seen, Rajsekhar.
Now not showing.
Not showing, sir.
Not showing the
No,
screen.
no, it's not
Now
seen.
it's okay, sir.
No,
No.
Oh, sir.
గోపి నమస్తే. ఆ రాజశేఖర్ స్క్రీన్ షేర్ అవ్వడం లేదు, ఒకసారి చూడు. వెళ్ళి చూడు. ఆయనకు చేస్తున్నాడు కానీ ఆయనకు రావడం లేదు, ఒకసారి చూడు. లేదంటే అన్షేర్ చేసి మళ్ళీ షేర్ చేయి. ఓకే.
She is failed to start playing letter. Okay, madam.
Okay, go ahead.
Okay, sir, continue.
This conventional biological indexing still remains very, very valuable and useful tools for the diagnosis and maintenance of these virus cultures. In biological indexing, graft inoculation. In biological indexing, graft inoculation or sap inoculation will be carried out. Specificity is very low to moderate. CTV in case of acid lime indicator plant actually take 1 to 2 months for symptom expression, whereas in case of citrus greening disease, citrus yellow disease, French bean, on sap inoculation, it take only 10 to 15 days for symptom expression. These are the limitations there with the biological indexing methods, but still these methods are very useful for the maintenance of the cultures and diagnosis. In Andhra Pradesh, citrus greening, citrus yellow mosaic virus, and citrus diseases are the major budwood transmitted diseases. These are produces very conspicuous symptoms on the plants. Even in some of the times, these diseases appear as a mixed infection. These are the mosaic symptoms appear on the different citrus species, sweet orange, acid lime. You see the very highly conspicuous symptoms. And pumela also is somewhat variegated symptoms. At citrus registration, these are the three major budwood transmitted diseases. Only citrus green disease, it is vectored by Diporna citrus psyllids, and it is around 36% disease incidence was observed in the Andhra Pradesh. Citrus yellow mosaic virus, Badana virus, is transmitted by the mealybugs. 35% incidence was observed in Andhra Pradesh. Whereas citrus disease virus is a cluster virus. It is a if transmitted virus. It is around 24% incidence was observed in the Andhra Pradesh. These are the major budwood transmitted diseases observed in the Andhra Pradesh. At citrus registration Tirupati, initially biological indexing methods were developed and used up to 2000 against the CTV, citrus yellow mosaic virus. During the 2001, molecular diagnostic laboratory was established at the citrus registration Tirupati with all essential equipments for diagnosis. So initially, this biological indexing, we used different citrus species for diagnosis of HLB. Among these species, citrus hystrix is got highest transmissibility. With this, we can diagnose the citrus greening disease. These are the different symptoms appear on these plant species. Later, polyclonal antibodies against CYMV were produced against the pathogen in rabbits. So actually, citrus illuminous virus is a DNA virus. This virus was partially purified, and this partially purified virus is injected to the rabbits for booster dose. The blood was collected from the rabbits, and from the blood, serum was collected. Later on, purified the antibodies from the serum. These antibodies are used against the diagnosis of citrus yellow mosaic virus. Actually, two methods are developed actually against this disease at citrus resistant Tirupati. One is a DAC ELISA and another dot immunobinding assay. These both techniques are developed against the citrus yellow mosaic virus disease. So with this, initially diagnosis the diseases at field level as well as in motherwood blocks, and used to eliminate the infected plants regularly. Later on, nucleic acid hybridization technique also developed at citrus resistant Tirupati. These are two probes are developed. So with this, up to 1:100 dilution of the tissue dilutions, we are able to diagnose this disease, as well as in case of DNA concentration around the sensitivity is around 100 nanogram per microliter. At CRS Tirupati, developed the PCR protocols against the citrus disease virus, citrus yellow mosaic virus, and citrus green disease. CTV, it is a feed-borne virus. It causes the stem splitting, vein clearing, stunting, and chlorosis. It also causes the slow or quick decline. Primers actually designed against the P25 and P23 regions of the say virus genome. This P25 is the actually a code protein region. It is a highly conserved region. In this region, we developed the primers. The product length is around 672 base pairs. In citrus breeding also, PCR protocols were developed. Actually, in case of citrus breeding disease, rRNA gene was used for diagnosis for primer design, and this rRNA gene product length is around 451 base pairs. Whereas in case of citrus yellow mosaic virus, again code protein used for primer design, and this is a conserved region used for primer design. Actually, we developed primers. This product length is around 726 base pairs. Later, standardized the duplex PCR for simultaneous detection of the citrus yellow mosaic virus and citrus green diseases. This duplex PCR, two primers actually designed against the 16S ribosyl RNA region and CP region of the citrus yellow mosaic virus. These both primers used at once, and we were able to diagnose the disease as mixed infections. So here you can clearly see the at sample 7, both bands are repaired. One is a size of 726 base pair, another is 457 base pairs. Indicates that both viruses are present in the plant sample. These are the duplex PCR developed and we are regularly using for the disease diagnosis. During 2016, under RPO project, established the laboratory with all facilities, actually real-time PCR. This is real-time PCR is a highly sensitive and reliable quantitative method for pathogen detection. In this here we can see observe the progress of the reaction in real time. There is no need for the fast PCR steps like post PCR steps like gel electrophoresis, gel documentation. There are two types of real-time PCR techniques are there. One is cybergreen method, another one is a probe-based method. Among these methods, probe-based method is somewhat sensitive. In that research station, we developed both these techniques, cybergreen as well as probe-based method. We able to standardize, quantify the virus load also in the plant samples. These are the plant samples infected with the viruses diagnosed at our research station. So what actually diagnostic methods like diagnostic disease like citrus yellow mosaic virus, citrus tister virus in HLB, and the Indian citrus ring spot virus developed. In actually citrus yellow mosaic virus, qPCR and duplex PCR methods were developed. Like citrus tister virus, DAC ELISA and RT-PCR techniques were developed. HLB, qPCR and duplex PCR techniques were developed. In case of Indian citrus ring spot virus, DAC ELISA protocols were developed for that diagnosis. For production of the disease-free planting material, there are 4 steps are there. One is a selection of the mother trees and indexing of the mother trees, re-indexing, establishment of foundation blocks, and multiplication. These are the steps involved in the production of the disease-free planting material. So it is a systematic representation of budwood certification in citrus. Actually, there are 2 starting material. One is the selection of superior local mother plants or imported budwood. These two, with these 2 starting material, the budwood certification starts. After selection of the superior mother plants, the propagation of the virus, propagation and virus indexing will be carried out. In case of imported bud material, there is a therapy or shoot tip grafting. These methods will follow to eliminate if any infections are there on the material, bud material. Later on, index material was established as a foundation blocks. These foundation blocks, again, virus indexing will be carried out. And again, foundation blocks at nursery also establishes. After raising the desirable rootstocks, insect-free net house, this virus-free cyan material packed from the foundation block at nursery, and large-scale propagation will be carried out under the net house. This is the production of certified plant material. This is a systematic representation. At our research station, we are following these methodologies for the production of the certified disease-free planting material. For selection of mother plants, initially at our research station, under the nuclear trees raised for selection of allied trees. Actually, these are all budwood transmitted diseases. If you select grafted material, there is a chance of infection. So hence we selected the these nuclear seeds. Why? Because these budwood transmitted diseases are not seed-borne diseases. Hence we selected the nuclear trees for raising of the mother blocks. Among these nuclear seeds, allied trees were selected. Promising mother trees were selected on the basis of yield, tree vigor, fruit quality parameters such as fruit weight, TSS, and tree health. Each one of these characters, 32 outstanding mother plants were finally selected for further multiplication in the greenhouse. The budwood was collected from the selected outstanding trees, propagated on Rangapur lime rootstocks, seedlings, and maintained on 110 plants in greenhouses. These are the old greenhouse where we maintain the 110 plants for collection of the bud material. Later, during the 2016, we were able to establish on a big greenhouse in the area around 4,000 meter square area at citrus station Tirupati, and one more is established at Mahanandi, HRS Mahanandi, 1,058 meter square area. These are the 2 mother blocks established at 2 different locations. Multiplication. So once the establishment of these foundation blocks at research stations, so we able to collect the disease-free buds from these plant, and we supplied around during 1996 to 1999 supplied 1 million buds to the nurseryman for propagation. From 1999 to 2009, around 22 lakh buds actually supplied to the nurseryman, and around 3 lakh 97,000 plants supplied to the farmers from the different regions of the South India. During the 2010 to 2020, the production of the plant material also doubled. They reached to 7 lakh material, and 1.4 million buds are supplied to the different nurserymen. This is the achievement during the last decade at our research station. So out of these research and projects, we actually recommend the disease-free planting material for raising the Sathgudi plants. And budwood mother plants were selected from the nuclear trees based on the horticulture characteristics. Trees were maintained in screen houses or indexed for budwood transmitted diseases such as yellow mosaic virus, CTV, and by DAC and PCR. These projects have supplied 3.72 million virus-free Sathgudi buds to the different stakeholders. Yearly, 0.6 to 1 lakh virus-free Sathgudi buddings budded on the Rangpur lime rootstock were supplied to the growers in South India. Around 560 Sathgudi sweet orange trees are being grown in for the budwood supply and for production of the virus-free buddings. Budwood trees being indexed regularly by RT-PCR, multiplex PCR against the budwood transmitted diseases like citrus trichia virus, citrus greening, and citrus yellow mosaic virus. An amount of 5 crores the sale of disease-free planting material of sweet orange Sathgudi. The center is responsible for establishment of more than 3,980 hectares of healthy orchards in the farmers' fields, predominantly in South India. These are the achievements. Future needs based on our success story of indexing and production of disease planting material at citrus Tirupati. The technology associated infrastructure need to be replicated at major citrus growing regions of India. There is a need to develop a diagnostic kits for the detection of these viruses. Need to take up periodically capacity building trainings for field functionaries, nurserymen, and technical staffs. Thank you. Any questions?
Thank you, Sir Rajsekhar Garu. It's a very informative and elaborative lecture, sir. Thank you, sir, for nice presentation. It is giving a very good assurance for the farmers to grow high yielding and disease-free plants. Sir, is there any possibility to collect the budwood from the gardens that are established from the farmer fields, sir, where these plants are established as a garden?
Madam, actually, all these budwood transmitted diseases are vector transmitted diseases, and already these diseases are established in the growing areas. Hence, we are not recommending to collect from those orchards. Why? Because there is a chance of reinfection on those disease-free plant material. Hence, before collecting the bud material, we should index those trees to confirm that there is no infection. So then we can collect the budwood material from those plants. But based on our experience, what we observed is 60 onwards, almost all plants are infected with these diseases. Hence, we are not recommending to collect from the unprotected orchards.
Okay, sir. Thank you. I request student participants to interact and to have any, if you have any queries. Sir, there is one question in chat box, sir. In our area, sir, there were very less fruiting I seen in Hanumangarh, Rajasthan, and also in Sirsa in Haryana. What is the reason and behind it?
See, it is mainly based on the environment conditions, especially actually citrus, you have to give some stress for the flowering. There is no stress during the flowering stage. Sometimes there is a delay in flowering may occur. It's mainly due to the see, in case of Andhra Pradesh, what we observed is last season there is a continuous rains. That's why there is a delay in the flowering. That is the main reason. Have to see what we have to give some stress to those plants for getting flower. Is the main reason. That is the main problem.
एक्सक्यूज मी सर, बारिश तो सर अब हुई है सर, पहले तो फ्लोरिंग के टाइम कोई बारिश नहीं थी। आए
आपके
साल
लिए-
होती है सर फ्लोरिंग।
अब कैसे
इस बार
इरिगेशन है आप गार्डन में?
इरिगेशन है सर,
इरिगेशन
प्रॉपर
फैसिलिटी।
इरिगेशन।
हाँ, ज्यादा इरिगेशन करने से भी एक प्रॉब्लम आता है, थोड़ा स्ट्रेस क्रिएट करना है फॉर।
अब क्या ट्रीटमेंट करें सर?
अब वेट करना पड़ेगा आपको फ्लावरिंग के लिए। अब बारिश हुई है ना?
यस सर, अभी तो हो गई बारिश।
हाँ, आपका फ्लावरिंग सीजन एक्चुअली एट आवर सेंटर एट योर स्टेट व्हाट इज द फ्लावरिंग टाइम?
फ्लोरिंग सीजन सर हो गया सर, फ्लोरिंग सीजन तो अब तो फ्रूट भी गए। नवंबर दिसंबर में फ्रूट आने लग जाते हैं सर।
हाँ हाँ, अब तो थोड़ा वेट करना पड़ेगा फ्लोरिंग के लिए, थोड़ा स्ट्रेस दीजिए, आएगा फ्लोरिंग।
ओके सर, थैंक यू सर।
थैंक यू।
There is one more question in the chat box, sir. What are nucellar seedlings?
Nucellar seedlings means from some seed. See, actually, in case of citrus, there is nucellar embryos, nucellar seeds and embryo seeds are both two are there. If the seedling is arised from the zygotic seed, that is called embryonic seed. The nucellar material, then it is a nucellar seed. So it is nucellar seed is a true type of the mother plant. Any horticulture person can answer properly. Then, madam.
Yeah, that is correct. What you said is correct.
Right.
Hello.
It's a true to type.
Yeah, madam, since we are propagating, especially this comes for acid lime and other lime and lemon groups. In order to get the true to type seedlings, we only prefer nucellar seedlings, which stands 2 to 2. Whether the zygotic seedlings are there, other type, they segregate. All the seedlings which come out of the single seed will not be same. So generally we separate at the nursery stage itself. We select the nucellar seedlings only in order to get the true to type planting material. This is basic thing.
Yes, sir. Thanks.
Any more questions?
Sir,
More questions are there, madam. The chat box.
sir, no, sir. Sir, now I request Associate Dean, sir.
Yes, madam. Yes. Are there any questions from student side? Or even the faculty can also clarify their doubts regarding this diagnosis, indexing. I think some of the staff members are also participated in this program. Okay, if there are no questions, no? Any questions from the student side or from the faculty? Okay, then thank you, Dr. Rajasekharam garu, for your nice presentation on today's topic. There is a budwood transmissible disease indexing in citrus for the production of disease-free planting material. The presentation was very good. because you have shown very good the photographs of viral diseases and diagnosis of different viral diseases, how the diseases transmits to the plants by different vectors, and different protocols, particularly the protocols developed by the institute for the diagnosis of major bloodwood transmissible diseases. And another important thing is, at the end of the presentation, you have told some 3.72 million and disease-free bloodwoods were supplied to different nursery people, and there was an earning of nearly 5 crore rupees. It is, I think, a noteworthy contribution from the citrus research station Tirupati. It is really one of the pioneer institutes working on citrus for the benefit of the citrus growers, not only in AP, but also our material has been, even the farmers from different states, Maharashtra, even Rajasthan also, some of the farmers have they have got the material from our research center. It is really very privileged institution for the production of disease-free planting material. And on behalf of our Honorable Vice Chancellor and the Registrar and all the university officers and co-conveners of this program and course directors, organizing committee and participants of today's program, I extend my heartfelt thanks to you for sparing your valuable time and presenting one of the wonderful topic on citrus. I think my colleague Dr. Ruth Garu, do you want to add some more points?
No, sir.
Okay. Okay. I think from all the participants' side, and also on my own behalf, I extend our heartfelt thanks to Dr. Rajasekharam Garu for presenting very excellent topic on citrus. And also we are also thankful to Dr. K. Gopal Garu, Registrar of Dr. Y.S. Horticulture University. Though he was not physically seen here and present here in this program, I think he was very much interested in this program to go with a grand success. And as I was told in the beginning, he was busy with some other engagement. That's why he could not able to attend this program. So in spite of his absence, I think Dr. Rajasekharam has fulfilled the presentation with very good information. I think it's really very useful presentation for the students as well as the faculty also. I once again extend my heartfelt thanks to Rajasekharam Garu for his wonderful presentation on today's topic. I think Swaraj Lakshmi Garu, do we have that program, quiz program? Or
yes, sir. Yes, sir. I will help Johan.
yes.
Actually,
Okay. Then
this is
I think that I will also. Sorry, Dr. Ruth also wanted to say something about this today's topic. I think you are also a pathologist. I think you can speak a lot about this today's program. Being a pathologist, senior most pathologist working in or horticulture university, I think you can also share your experiences on today's topic and say a few words to Rajasekharam Garu for sparing his valuable time in giving an excellent talk. May I request Dr. Ruth Garu to kindly say a few words about this program.
Sir, these viral diseases, especially citrus greening, is the most dangerous disease in case of acid lime, sir, and sweet orange. By using these disease indexing, we can supply this planting material by using these indexing techniques. It is a great achievement to the citrus research station by the this is developed by our registrar sir and K.T. Ramana sir. Developed a citrus bud bank at Mahanandi Citrus Horticultural Research Station also. I developed some 6 acres citrus bud bank at Horticultural Research Station Mahanandi for the supply of this planting material, sir. Thank you, sir.
Yes, may I now request Dr. Ramana, who is also an eminent scientist who has worked in Citrus Research Station Tirupati. I think he has also made some significant contribution for the development of citrus industry in AP. And he was there for a long period at Citrus Research Station. And he is also part of the development of Citrus Research Station and also the production of disease-free planting material and supply of planting material to farmers in different states. He is also one of the unsung heroes in the developmental process of Citrus Research Station. I think it is a hereditary. I think we can see such a wonderful scientist who have already worked in the institution, and the presently Dr. Nagaraju and his colleagues who are there at the Citrus Institute, they are doing excellent work. I think I request Dr. Ramana also to kindly say a few words on today's talk.
Thank you, Dr. Sinu. First of all, I would say one thing. This is the only center in the entire South India where indexing works against these bacterial and viral diseases are carried out, especially in citrus. We have 4 to 5 very prominent centers under ASAP and also at one National Institute IHR. But when we talk about citrus, this is the main center even in ASAP also, which is recognized well for this indexing works. And because of that, the research station has been producing, we are continuously doing that indexing for CYMB, CMB, and also greening. So during 2016, we got established the greenhouse, which was very much in need. Earlier it was not there, to protect the sweet orange mother plants from infection by this viral and bacterial diseases. We all know that greening is spreading very fast in the state. Earlier it was, we are not concentrating much on the previous diseases, age-old diseases like Tistija and all those things, but only 3 major diseases we are concentrating, which are prevalent everywhere, not only in AP, as well in AP, that is Telangana also. So major complaints are there as far as the viral and bacterial diseases are there. Of course, fungal diseases are also there. We are also doing some work on soil-borne fungal diseases also in citrus and acid lime especially. Many trials are under progress, and we are happy to say that this station has supplied a lot of disease-free planting material to the farmers of AP and Telangana, and also Karnataka and parts of Tamil Nadu also. And because of this recognition, the center has entered from the university side to the MOU with the private organization in Tamil Nadu, that is Tirunelveli district. So this is one major nodal center in ASRP for citrus. Whoever wants any information with regards to the citrus in the ASRP system also, from South Indian side, we are first touched upon. Citrus resuscitation is first asked for that, any sort of clarity. So that kind of good impression in this station is has even today. And I think Rajasekhar has, after Dr. Gopal, is Rajasekhar is continuing all his work in a very efficient way. And also not only plant pathology, even entomological aspects, horticulture aspects also they are going on well there. In this regard, I compliment and appreciate all the staff working there for their good efforts in keeping up the name of the research station. Thank you, sir. Thank you.
Thank you, Dr. Ramana, for sharing your experiences with the citrus research station, where you have worked for quite a long period. And we must, as you said, I think compliment goes from all our side also to the scientists working there at the citrus research station for their wonderful work. We wish that the work should continue there for the benefit of the citrus growers, and not only in AP state, as you said, in other South Indian states also, where the citrus is, is becoming one of the important fruit crops, which is highly economical, and also nowadays it is fetching a high price in the national market. I think many farmers are now, as we have seen, most of the farmers are now going for the citrus crop by removing other fruit crops. I think there is a great contribution from citrus research station, and it should continue also, keeping in mind the importance of citrus crop. Thank you, Ramana, for sharing your thoughts and ideas for the benefit of the students and staff. Dr. Swaraj Rashmi garu.
Yes, sir. It's a very good platform, sir, for us to, we are able to see all screen houses that are developed for established for production of disease-free planting material. Where all participants are excited to see all the even the laboratory that is established at CRS for this work, sir. We have to congratulate and we have to thank our guest speaker today and all the team, Gopal sir, Ramana sir, and Nagaraj sir and their team, for giving us this opportunity and sparing their valuable time for giving us lot of information. On behalf of all the participants, we have to, it's our pleasure to extend our thanks to the guest speaker today, Dr. Rajasekhar garu and Dr. Gopal sir, Registrar Dr. Rajasekharam, for providing us this opportunity, for giving us a informative lecture on this platform. It is also my pleasure to extend my thanks to Dr. T. Janakiram garu, Vice Chancellor Dr. Rajasekharam, for providing us this type of opportunity to conduct this type of program, this citrus graduation readiness program for the benefit of agri-horticulture students, and also all university officers for giving us their support in conducting this program, sir. It's a nice program, sir. We are all thankful to you. On behalf of Associate Dean COH ARPETA and all student participants, we are extending our thanks to all university officers and guest speaker today for successful conducting of this 20th session, sir. Sir, I request Johan to load this questionnaire so that they will fill up and send. All participants can fill up and send. Is it possible now, Johan?
I think Johan is there.
Sir, today is the 20th session.
Yes.
That's why we are giving the second test for all the participants today.
Madam,
The same test will be.
Madam, I sent already the link into the chat box, madam.
Okay, Johan, thank you. All the staff who are supporting us in conducting this citrus graduation sessions, and also especially for today's session, all the staff members, I thank immensely for their support for conducting this session. Thank you, one and all.
Thank you, madam. Thank you, Dr. Rajasekharam. Thank you, Dr. Ramana. Thank you.
Thank you, sir. Thank you for giving me this opportunity.
Thank you very much. Dr. Ruth Garu and all, Dr. Surajalakshmi and all other faculty who have participated in this program, and dear students, make use of this program. This is a wonderful program that is being organized by Dr. YSRHU for the benefit of the final year students. I once again request all the students who are in ELP program, please make use of this program. Though you may be engaged in, in your activities, daily activities, you kindly see that your participation must be there in this program also, because 10 more lectures are yet to, I think, to come in this Citrus Graduate Readiness Program. So far, we have completed 20 lectures. 10 are yet to come. I request all the, staff members also, faculty, and the course directors, co-conveners of this program, and also the committee members, all the other committee members, organizing committee members also requested to kindly see that the students' participation will be there in this, in the next coming lectures. Thank you very much to all of you.
Thank you, sir.
Thank you.
Thank you.
I think we can close this program. That already the questionnaire is there in the chat box. No? Students can.
Yes, sir. We will wait for 2 minutes, sir.
Okay. I think if there are any difficulty in going through that chat box and in filling the answers, I think students can clarify their doubts from Dr. Johan. Dr. Johan? Johan?
Sir.
Now, have you placed that questionnaire in that chat box?
Yes, sir. I put it the chat box and our group also, sir.
Okay. If there are any queries from the student side in the chat box, how to open that, how to fill that answers.
No problem, sir. It's very easy, sir. Very easy, sir.
Because already we have once we have given that.
Yes, sir. First they have to give their information, sir, like what is the name and ID number and the college and university.
Okay.
Then they will going to the next. They can submit the next. They can press the next option, automatically entering into the examination, sir.
Okay. if there are no doubts from the student side, I think we can end this program. Swarajyalakshmi garu. Dr. Ruth garu. Swarajyalakshmi garu, I think Dr. Johan has done
Yes, sir.
whatever necessary. He has already posted that questionnaire in the chat box.
Okay.
I think if there are any doubts, we can
It will
clarify
take
further.
another 5 minutes time, I think, sir, for the students to fill the
Okay.
answers. It will take another 5 minutes, sir. After 5 minutes, we will close, sir. We will
Okay,
give
okay.
5 minutes time for the students to participate.
Okay. I think if there are any suggestions from our side, you can just explain them. So within that 5 minutes time. I think others, if there is some other activity, I think they may leave this program. Other participants, if there are any other important activity.
Yes.
Yes.
Yes, sir. Those who are filling the answers, they can take another 2-3 minutes time.
Okay.
I request all the participants to inform to your friends also, or students, all the students to inform to your friends also to participate in the next coming 10 sessions, at least without fail. Participate in all the programs, as this is the good opportunity given to you all. It's a nice platform to learn many things about citrus, its production technology, improvement, then you may take another 5 minutes time. It's a Benevol, you can take another, I think it's 10 minutes, overall 10 minutes time, I think, for filling up of all the questions. Johan, how much time, Johan, for
Madam, 10 minutes, madam.
okay.
So Rajalakshmi garu, there is one request from Rishabh Beniwal.
Now which request? I will see, sir. I will see, sir.
Please
Ma'am,
respond to
please
him.
respond about I don't show previous test marks. Please tell. We will send it to you, Rishabh.
Then
You can see you score will come to you. You can see your score.
Madam, first test result I already sent to him, sir.
First test results already it was sent to you, I think. Again, we will send. Johan, if it is required again, you please send to him.
I already sent to him,
Okay.
sir. I already sent to him, madam.
Okay, okay.
So Rishabh Beniwal is one student, wonderful
Regularly
student.
attending.
Yes, he is very sincere and regular. I think
Yes,
he is seen in every program.
yes, sir.
Yes, we must all congratulate him.
Yes, sir.
I think he will.
Thank
This
you,
will
sir.
definitely. This program will definitely give him a big boost in his career.
Yes, yes, sir.
I think
Thank you, sir.
We should all wish him that he should come up very well in his
Yes, sir.
future studies and his career.
మన స్టూడెంట్స్ అంతా పార్టిసిపేట్ చేశారా లేదండి? జోస్నా, రాజేశ్వరి, కిషోర్ నాయక్,
Yes,
Nanda
yes.
Kishore. Have you attempted the test today? India.
Still only 3 members are attended, madam.
Why? What about others? The number of students are here. Rania Sherin, Kishore Nayak, Nanda Kishore from Tadipatri, I think, Nanda Kishore. And Priya.
Actually,
Please
ma'am,
attend. This type of test, it will give you some experience. How to answer the questions. Time management also, you will be knowing
Madam, only
by
this
answering.
person, madam, only this persons are submitted their exam test.
How
Only
many? How
3 or
many?
4.
How many, Dr.
3 or
Evan?
4
How many?
members.
How many students have participated?
Sir, around 20-25 students are here.
Okay.
But only attended only 4-5 members only attended.
For
నేను కూడా
this
పంపించా ఈరోజు.
quiz program, for this quiz program. Only
Why students? No.
Yes, sir. This is the
It's a
quiz
very-
program result, sir.
Okay.
It's only
Okay,
funny thing that you can learn how to answer the questions and time management also. You will be learning.
Okay.
స్టూడెంట్స్ అయితే ఉన్నారు సార్ చాలా మంది, కానీ ఎందుకు పార్టిసిపేట్ చేయడం లేదు? వీళ్ళు ఆన్సర్ ఓపెన్ చేసి లింక్ ఓపెన్ చేసి ఆన్సర్ చేయడానికి ఏంటి ప్రాబ్లం?
Yes. That is the doubt we are getting, whether they have any really problem to.
सर प्रॉब्लम ये नहीं है सर, प्रॉब्लम ये है कि ऑनलाइन में ज्वाइन होके सो जाते हैं बच्चे।
हो
I
जाते
see.
हैं।
Okay.
Mr. Rishabh,
Yes, sir.
I have seen your question in chat box. You are from Allahabad to UP.
No, sir. I am from Mangadh, Rajasthan. I am
Rajasthan.
studying
But you are doing your BSc in UP.
Swatsilavad.
Okay. Now the question is your Rajasthan. What is the crop? There is a poor, very less fruit setting is seen. Which is the crop?
Sir,
You don't have
Kinnu.
Yes, Mandarin. Which is the major season? Which season you get the very less fruit setting?
सर इसी साल हुआ है, नवंबर दिसंबर में फ्रूटिंग आती है।
No, वो तो एम्बिया क्रॉप है, पता है मुझको। एम्बिया क्रॉप मेजर क्रॉप इज गंगानगर एरिया, राजस्थान, पंजाब, दे भी वो एम्बिया क्रॉप ही लेते हैं। लेकिन इस साल की yield कम किस लिए है? How is the temperatures there? Because it is very cold during winters.
Yes, sir. Cold तो बहुत है इस बार.
This is happening, and even before year also there was less fruit setting, and you get the hailstorms also during the now December to April, and there is a heavy loss also crop. And the second one is Sirsa in Haryana. How do you know the Sirsa area in Haryana?
Sir, बिल्कुल पास में ही हमारा गांव है sir.
Okay, it's a border to your state, isn't it? Huh? But Haryana, they don't have citrus much.
Yes sir, है sir. मेरे नाना जी खुद का बाग है.
अच्छा, कौन सा वो वहाँ क्या है? वो भी मैंड्रिन है?
Yes sir, किन्नू ही लगाते हैं.
हाँ, किन्नू. और ठीक है. क्या एज कितना है ट्रीज का?
5 years.
5 years is very early.
Sir,
5 years is very early.
सर जो पुराने हैं उनके भी नहीं हुई तो वहां तो.
हाँ, वो पुराना कहाँ है? राजस्थान में या सिरसा में?
सिरसा में भी पुराना है और भी वहाँ मेरे नाना जी के आसपास
हो
बाग.
सकता है इस बार थोड़ा टेम्परेचर वेरी माइल्ड है, मे बी फ्लावरिंग होगा लेकिन लेट. ओके, तो आप इतना 2-3 स्टेट में है इसलिए बात मेरे को लगा है वो मैंड्रिन होगा, क्योंकि वहाँ नॉर्थ में तो स्वीट ऑरेंज नहीं रहेगा और एसिड लाइम भी.
सर मेरे ज्यादा स्टडी नहीं किया हुआ क्योंकि मैं फर्स्ट ईयर का स्टूडेंट हूँ.
अच्छा, ठीक है, कोई बात नहीं. लेकिन आप एक बार वो गंगानगर या लुधियाना अबोहर सेंटर से अबोहर ले लेना, आपका बॉर्डर में है स्टेट का वो. सो यू विल गेट द करेक्ट पिक्चर. ओके, वो भी हमारा सेंटर है, तो दे आर वर्किंग ऑन सेम किन्नौ.
Yes sir.
Okay, try to get their numbers and contact, you will get the correct information.
Okay sir.
Okay, Nisha.
Thank you sir.
Hello. Good evening, madam. Cheppandi.
Madam, shall I end the session?
Yes, Johan.
Shall I end the session, madam?
Yes, Johan.
Okay, madam.
Now we are concluding the session today. Our next session is on 24th.